Dear students, Assalamu alaikum. In this lecture video, I shall try to discuss about protein. Hope this video will be helpful for you all. After publishing my previous lecture video, some of you suggested me to deliver my lecture in mixed language, that is, both in English and Bangla. From this lecture video, I shall try to deliver my lecture using both Bangla and English. At first, if I ask what is protein, I know everybody will answer the same thing because all of you read about protein in your higher secondary level. Protein are the long chain polymers of amino acid. Different type of amino acid join with each other to form a long chain polymer, that is protein. As we already said that protein is a polymer, now we have to know what is polymer. A polymer is a long chain molecule that is formed from small molecules which are known as monomers linking together. Polymers are made from monomer. Polymers are two types, homopolymer and heteropolymer. Homopolymer is a type of polymer that is made from identical monomers, and heteropolymer is a type of polymer that is made of two or more different types of monomer. We can say starch as a homopolymer because we know that starch is made of many glucose molecule only. And we can say protein as a heteropolymer because protein is made by different amino acid. In our body, different types of proteins are present, but chemically all proteins are the polymer of amino acid. But every protein has unique sequence of amino acid. That's why their function is different from each other. Here we discuss about different classes of protein according to the function. So according to the function of protein, we classify protein in the following classes. We will discuss the class of protein step by step. First one is structural protein. Actually, the name is related to the function. Which proteins are known as structural protein? Those proteins provide structural components in our body. They are known as structural protein. For example, keratin. Keratin is a protein which is present in our hair, skin, wool and nails. As they provide structural components, that's why they are known as structural protein. Next one is contractile protein. Contractile proteins are those proteins which are involved in the movement of muscle. For example, myosin and actin. We know myosin and actin contract muscle fibers. That's why they are contractile protein. After that, we'll discuss about the transport protein. Transport protein are those protein which carry essential substances throughout the body. For example, hemoglobin. We know in hemoglobin has two parts. One is heme and another part is globin. Heme is iron and globin is protein. Actually, hemoglobin can transport oxygen through blood in our body. That's why hemoglobin is known as transport protein. Next class is storage protein. Storage proteins are those proteins which can store nutrients in our body. For example, ferritin. Ferritin is a kind of protein which can store iron in our bodies, particularly in liver and spleen. That's why it is known as storage protein. Next class is hormone. Hormone is a protein which can regulate body metabolism and nervous system. For example, insulin. We know insulin can regulate our blood glucose level. That's why insulin is known as hormone. Next class of the protein is enzyme. Enzymes are special proteins which can catalyze biochemical reactions in the cell. For example, sucrase. Sucrase is an enzyme which can catalyze the hydrolysis of sucrose. That's why sucrase is known as enzyme. The last class of this protein is protection protein. Protection protein are those proteins which can recognize and destroy foreign substances in our body. For example, immunoglobulins. Immunoglobulins stimulate immune response in our body. That's why this protein is known as protection protein. I already told that there are different types of protein in human body, but all proteins are polymer of amino acid. They differ in amino acid sequence only. In the previous slide, we discussed about the classification of proteins based on their function in human body. Here we classify protein in a broader sense, that is broadly we can classify protein in two classes. One is fibrous protein and another is globular protein. Actually, fibrous proteins are the structural proteins, that is those proteins are used for structural purposes in our body. But remaining all proteins which are used for non-structural purposes are known as globular protein. Fibrous proteins are insoluble in water because they are made by those amino acids which are insoluble in water. But globular proteins are more or less soluble in water because they are made by both type of amino acid, that is soluble amino acid and insoluble amino acids. That's why they are more or less soluble in water. We'll discuss about different types of amino acid in next slide. For understanding the chemistry of protein, you have to know about the chemistry of amino acids. Amino acids are the building block of protein. Here we can see a common structure of amino acid. Amino acid contain a carboxylic acid group and a amino group on the same carbon, and it contains another hydrogen and another side chain because carbon can form 4 bonds. Proteins are composed of these amino acids. Amino acids are sometimes known as alpha amino acid because in case of carboxylic acid, we know that the next carbon of the carboxylic group is alpha carbon. In case of amino acid, the alpha carbon of amino acid contain a amino group. That's why it is known as alpha amino acid. In aqueous solution, amino acid form a bipolar ion. That is, it contains a positive charge and a negative charge. This is the special characteristics of amino acid. As amino acid form a bipolar ion in aqueous solution, and this bipolar ion is known as Zwitter ion. A bipolar ion or Zwitter ion are those ions where both positive and negative charge was present on the same molecule and the net charge of the same molecule is 0. How amino acid form bipolar ion or Zwitter ion? In the structure of amino acid, we already saw that there is an acidic group and a basic group. We know that acids donate proton and bases accept proton. In aqueous solution, the acidic group of amino acid donate a proton and form a carboxylate anion. After that, the amino group of amino acid accept that proton from the solution, because the nitrogen of amino group is a basic group and it has a lone pair of electron. Using its lone pair of electron, it formed a bond with this proton. For forming this bond, both electrons were shared by nitrogen. That's why the charge of the nitrogen become positive. This way, amino acid form a bipolar ion, that is Zwitter ion. Here we can see the structure of two amino acid, that is glycine and alanine. The difference between these two amino acid is only in the side chain. Actually, one amino acid differ with another amino acid only in the side chain. And in the right side, we showed the 3D structure of glycine and alanine. Here the stick represents the bond and ball represents the atom. Actually, we are showing this 3D model of these two amino acids to inform you that in nature all chemical substance exist in 3 dimension. But when we write or draw the chemical structure of that compound, we write in 2 dimension, because it is difficult to write or draw the exact bond length and bond angle. That's why we could not write or draw the 3D structure of a particular compound. But nowadays there are many software like CamDraw, ISysDraw. By those software we can draw the 3D structure of a particular compound. Now I shall discuss about the classification of amino acid. We already know that structurally one amino acid differ from another amino acid only in the side chain. Depending on the nature of the side chain, amino acids are classified into 4 classes: non-polar amino acids, polar amino acids, acidic amino acids, basic amino acids. Non-polar amino acids are those amino acids which amino acid has a non-polar side chain. For example, alanine. We can see from the structure that alanine contains a methyl group, that is a non-polar group, as side chain. And we already know that non-polar groups are hydrophobic in nature. That's why non-polar amino acids are also hydrophobic in nature. Next, polar amino acids. Polar amino acids are those amino acids which have a polar side chain in its structure. For example, serine. We can see there is a polar side chain in serine. And we already know that polar groups are hydrophilic in nature. As a polar amino acid contain a polar side chain, that's why that amino acid is hydrophilic in nature. Acidic amino acids. Acidic amino acids are those amino acids which has a extra acidic side chain. For example, aspartic acid. We can see there is an extra acidic group, that is carboxylic group, in the side chain of aspartic acid. As we know that carboxylic groups are hydrophilic in nature. That's why acidic amino acids are hydrophilic in nature. Next, and the last one, is basic amino acids. Basic amino acids are those amino acids which have a basic group, that is amino group, in the side chain. For example, lysine. We can see there is a amino group in the side chain of lysine. That's why lysine is a basic amino acid. And we also know that basic groups are also hydrophilic in nature. That's why overall basic amino acids are also hydrophilic in nature. Among the 4 classes of amino acid, only non-polar amino acids are hydrophobic in nature, but the remaining 3 classes are hydrophilic in nature. Actually, the fibrous protein or structural proteins are made of these non-polar amino acids. That's why structural or fibrous proteins are non-polar, that is water insoluble. But globular proteins are made of these 4 types of amino acids. That's why they are more or less water soluble. There are many amino acids in nature, but only 20 different amino acids are present in the protein of human. Among the 20 amino acids which are present in human protein, 9 are non-polar amino acids. We can see the structure of the 9 non-polar amino acids in this slide, and we can see the difference is only in the side chain, and all side chains are non-polar in nature. 6 amino acids are polar amino acids. We can see the 6 polar amino acids in this slide, and we can also see in the structure that the side chain of these polar amino acids are polar in nature. 2 amino acids are acidic amino acid and 3 are basic amino acids. Acidic amino acid contain a acidic side chain and basic amino acid contain a basic side chain. These are the 20 amino acids which are present in human protein. Among them, 9 are non-polar, 6 are polar, 2 are acidic and 3 are basic. In this slide, we can see something is written for every amino acid within the first bracket. Actually, these are the symbol of that particular amino acid. Actually, these symbols are used to write the sequence of a particular protein. We can see 2 symbols for every amino acid. One is single alphabet symbol and another is 3 alphabet symbol. Actually, for sequence writing, everybody prefer the 3 alphabet symbol, but we know that if we use the single alphabet symbol, it will occupy less space. After that also we prefer 3 alphabet symbol for writing the sequence of protein. Why? Because for a single alphabet symbol, usually the first alphabet of a particular amino acid is written as single alphabet symbol. But problem is that there are many amino acids which start with the same alphabet. That's why for other amino acid, different alphabet was used as single alphabet symbol. But in case of 3 alphabet symbol, the first 3 alphabet of a particular amino acid is used as its symbol. First one is capital and the second and third one is in small letter. So using these 3 alphabet symbol, we can write the sequence of a protein or the amino acids in a protein like this way. This one symbol, then hyphen, another amino acid symbol, then hyphen, another amino acid symbol, like this way. By single alphabet symbol also we can write the sequence, but I already told that we prefer there are 3 alphabet symbol system. Now question is that in 3 alphabet symbol, is there any possibility to overlap the 3 alphabet with the another amino acid symbol? Yes, this also happen in case of this 20 amino acid. Aspartic acid's 3 alphabet symbol is ASP. Glutamic acid's 3 alphabet symbol is GLU. It overlap with aspargine and glutamine. Aspargine, in case of aspargine, the first three alphabet is ASP. In case of glutamine, the first three alphabet is same as the glutamic acid. But in case of aspargine and glutamine, if we pronounce aspargine and glutamine, there is a excess emphasis on N when we pronounce these two words. As this three alphabet overlap with this and this three alphabet overlap with this, that's why in case of aspargine and glutamine, the third alphabet was replaced by N. That is, for aspargine the symbol is ASN, and for glutamine the symbol is GLN. We can easily remember this symbol because for pronounce aspargine and glutamine we have to give emphasis on N. Actually everybody prefer three alphabet symbol because it is easy to remember the three alphabet symbol. That's why we prefer three alphabet symbol to write the sequence of amino acid in a protein chain. Now I shall discuss about the Fischer projection of amino acid. The Fischer projection was devised by German chemist Hermann Emil Fischer in 1891, which is a two-dimensional representation of a three-dimensional organic molecule by projection. Fischer projections were originally proposed for the depiction of carbohydrates, that is the monosaccharides, and used by chemists particularly in organic chemistry and biochemistry. They can also be used for amino acids or for other organic molecules. According to Fischer projection, we can draw the two-dimensional structure of amino acid. But from that two-dimensional structure, we can imagine the three-dimensional view of amino acid. For this, we have to draw a plus sign for a amino acid, and this connecting point represents the central carbon of amino acid, and this 4 line is the 4 bond which is connected to different atom or functional groups with the central carbon, because the carbon of amino acid can form 4 bonds. In the vertical line, we have to write the carboxylic group in the upper position, and in the lower position we have to write the side chain, and in the horizontal line we have to write the hydrogen and amino group. We already saw that the central carbon of amino acid is bonded with 4 different types of atom or groups. That means the central carbon of amino acids are chiral. Actually, all of the amino acids are chiral except glycine. In organic chemistry, we know that if an organic molecule contains a chiral carbon, that means it has 2 configuration. According to Fischer projection, these 2 isomer of that compound is known as D isomer and L isomer. That means every amino acid exists in 2 form. One is D form and another is L form. According to Fischer projection, we can distinguish these 2 isomer. If the amino group of a particular amino acid in the horizontal line is in the right side, in that case that isomer is D isomer. If the amino group is in the left side, that amino acid is L isomer. One can confuse this D and L isomer with the optical isomer. Actually, in case of optical isomer, we use small d, small l in italic. But here we use capital D and capital L for the naming of these two isomer according to Fischer projection. Actually, D isomer is the mirror image of the L isomer, and one is enantiomer to the another isomer. So we can see that every amino acid exists in two isomeric form, that is in two enantiomeric form. One is D isomer and another is L isomer. We can see here D alanine and L alanine both are mirror image of each other, and also D cysteine and L cysteine, these are also the mirror image of each other. Till now we came to know how to draw the two-dimensional structure of amino acid, how to distinguish the two enantiomers of amino acid by Fischer projection. But we did not get any idea about the three-dimensional image of amino acid. Actually, according to Fischer projection, the horizontal lines of the structure of amino acid usually written by bold line, and the vertical lines are written by bold dashed line. According to Fischer projection, this bold horizontal line represents that these two bonds projects towards the viewer, and the dashed line projects that these two lines are away from the viewer. That means in amino acid, if we draw the structure by Fischer projection, in that case the amino group and hydrogen atom projects towards the viewer, while the carboxylic group and the side chain projects away from the viewer. By this way, we can get an image of the three-dimensional structure of a amino acid by Fischer projection. We already know that 20 amino acids are involved in human protein formation. Now anybody can assume that every amino acid has two form, that is one is D form and another is L form. That means 40 forms of 20 amino acids are involved in human protein. Actually not this. Only L isomer of every amino acids are involved in human protein formation. Now anybody can ask that means D isomers are not present in human or in nature. Actually, D isomers are not involved in human protein formation, but they are available in other system in nature. That's all for today. I will discuss about the remaining portion of this chapter in next lecture videos. If you like my lecture video, then don't forget to click the like button. You can also share this video to your friends and can also put your comments. Lastly, don't forget to subscribe my channel. Your subscription will encourage me to make more lecture videos for you all. Thank you.